Good morning, students. Today, let us discuss about enzymes which are involved in DNA replication. Before that, let us discuss a little bit about the structure of DNA. According to Watson and Crick model of DNA is made up of two polynucleotide strands. The two strands are attached to each other by hydrogen bonds. The DNA molecule remains spirally coiled. A DNA molecule is made up of nucleotides. Let us see the structure of nucleotide. A nucleotide is made up of nitrogenous bases, deoxyribose sugar, and phosphate. The nitrogenous bases which are present in a DNA structure are adenine, thymine, guanine, and cytosine, where adenine always pairs up with thymine and thymine with adenine, cytosine with guanine and guanine with cytosine. Adenine forms a double bond with thymine, cytosine forms triple bond with guanine. Next, deoxyribose sugar. This is the structure of a deoxyribose sugar. Here we have seen that a hydroxyl group is replaced by hydrogen. So this makes the structure more stable. This is the phosphate. Now let us see the structure of a nucleotide, how these three components bind to form this structure. So this is the phosphate, this is the deoxyribose sugar. This is one of the nitrogenous base and this is the phosphate molecule. So here this is the carbon 1 and this is the nitrogen 9. Here it is carbon 5 and the phosphate molecule attaches with the carbon here. So this is how a nucleotide is formed. So several nucleotides combine to form a polynucleotide chain. So this is one nucleotide, this is another nucleotide, and they both are bonded with each other at 3, carbon atom 3 and here carbon atom 5. You can see the bond. The bond which is formed is known as phosphodiester bonds. Next, here we can see that the carbon 5 of this nucleotide is free. This is not attached with any other nucleotide. So this end it is known as 5' end, or you can write 5 up small dash 5'. Whereas here also we can see that the carbon 3, the hydroxyl group of carbon 3 is free from any bondage. It is not binded with any nucleotide. So this end is free. So this end is known as 3' end or 3'. The two polynucleotide strands, they are anti-parallel to each other. Here, if this strand runs to this direction, then this strand will run to the opposite direction. So they are parallel to each other, but they are anti-parallel. So here we can write 3' to 5' direction and 5' to 3'. DNA replication takes place from 5' to 3' direction. That is 5' to 3' direction. Now Let us discuss about the enzymes which are involved in DNA replication. The first enzyme which is involved is endonuclease enzyme. So endonuclease enzymes are those enzymes which can cut a DNA anywhere in the middle, not the terminals, but anywhere in the middle. So the first enzyme which comes into action in DNA replication is an endonuclease enzyme. This enzyme acts on one of the two sides of phosphodiester linkages which are present within the interior of a polynucleotide chain. In a double-stranded DNA molecule, it will cause a nick or incision. Okay, that is, the DNA will be present in one piece as you can see here, but a gap is created having two free ends. The next enzyme is helicases. DNA helicases are ATP-dependent unwinding enzymes which promote separation of two polynucleotide strands, thereby establishing a replication force that will progressively move away from the origin. So this is the nick or incision created by the endonuclease enzyme. Through this side, the DNA helicase enzyme will enter. Here we can see it is attached with the DNA duplex. Now it has started its action, that is opening the two DNA strands. Here we can see. So the entire strand is not open, but it opens slowly. The enzyme slowly moves forward, thereby opening the two DNA strands. Next, helix destabilizing strand. After the two strands are separated, they have the tendency to join back. So how this is prevented? This is prevented by a helix destabilizing protein known as single-stranded DNA binding protein. They bind with the newly formed exposed DNA strands without covering the bases, which therefore remain available for the templating process. Next enzyme is primase. Primase enzyme help in the formation of a short RNA primer strand. Here we can see this is the lagging strand. Why lagging strand? Because here the formation of DNA strand is continuous. Whereas this is the lagging strand because here we can see that the DNA cannot be formed from 5' to 3' direction. So it should be from this side like this. So here several short primers are established from 5' to 3' direction. So this is the lagging strand and this is the leading strand. Here it is continuous. For the formation of DNA strand is continuous, whereas here the formation of DNA strand is not continuous. So primase help in the formation of small RNA primers, RNA strands. They are known as RNA primers. These RNA primers, why these RNA primers are required? Because the next enzyme, that is the main enzyme which help in the formation of the DNA strand, requires an existing strand, polynucleotide strand for its function. Otherwise, it won't be able to lay down the nucleotides. Here you can see the next enzyme is the DNA polymerase enzyme. The DNA polymerase enzyme further lay down the DNA strand. The nucleotides are brought to its respective, the complementary nucleotides are brought to its respective sites, thereby forming a long strand here. So DNA polymerase enzyme require an existing strand so in order to lay down the nucleotides. So in leading strand, the formation is continuous as you can see here. Whereas in the lagging strand, I already told you, several small RNA primers are formed from 5' to 3' direction. So several small fragments of DNA are formed in the lagging strand by DNA polymerase III enzymes. These fragments, small fragments which are formed, they are known as Okazaki fragments. In this strand, the enzyme which is involved is primase for the formation of RNA primer. Here it is primosome, that is primase and DNA helicase both work together to form the RNA primers in the lagging strand. So we have discussed about the DNA polymerase III. Now let us go to the next enzyme, that is DNA polymerase II enzyme. Now, after the laying down of DNA strands, still the RNA primer is there. It is a DNA replication. So in a DNA strands should be there, not RNA strands. So what happened? Another enzyme, DNA polymerase II, binds with the strand, cut the RNA primers and laid down DNA nucleotides here. In lagging strand also, these small nucleotide strands, RNA strands are removed and new DNA strands are formed. Now, in the lagging strand, little gaps are formed even after the RNA strands are replaced by the DNA strand. So these gaps are sealed by another enzyme which is known as DNA ligase. Here, DNA ligase enzyme. Thank you.